Okay, I'm going to go ahead and introduce Greg. So Greg Silsbe is talking to us today. Greg is a research assistant professor at Horn Point Laboratory. He got his undergraduate in civil and environmental engineering at Queen's in Canada, and his master's and PhD in biology at Waterloo. He did a postdoc in the Netherlands Institute of Sea Research and a second postdoc at Oregon State University. And today, Greg is going to talk to us about PhytoChop. Go ahead, Greg.
All right, thanks very much, Clara. So good morning, everyone, and thank you so much for attending. I'd like to begin by acknowledging my collaborators on this project, shown here in these pictures, from Horn Point Lab, Sarah Malkin, Matt Gray, and Jamie Pearson, and former HPL postdoc Emily Brownlee, who is now faculty across the Bay at St. Mary's University College. They've been instrumental, pun intended, in getting this project up and running. I'll also note that Sarah came up with the name PhytoChop, which is an amalgam of phytoplankton and chop tank. And one of Jamie Pearson's creative pandemic outlets was designing our wonderful logo, seen here at the top of our slide, which is also available in sticker format. I have a really large stack in my office, so if you want one, please stop by. See if I can advance slides here. Here we go. So for context going forward, I need to also acknowledge that this is an NSF-funded project through the Biological Field Stations and Marine Laboratories, or FSML program, which is now within the larger infrastructure capacity for Biological Research Division. It's moved around a little bit. The scope of this program is to fund infrastructure that advances biological sciences, and proposals are judged by their ability to produce quality products that are openly accessible to a broad scientific community and education community that serves researchers beyond a single research team. In the presentation, I've underlined this last point for a couple reasons. First, just to admit that in our first attempt at this grant, I failed to fully address this criteria and we didn't get funded. Fortunately, this was a pretty easy mistake to fix, and so our second submission to FSML was funded, which is what I'm talking about today. The second and more important reason why I've highlighted this text is to point out that everyone listening to this talk constitutes this community of researchers. Simply stated, myself and my collaborators are the caretakers of the observatory, but the data products can and really should be used by as many people as possible. This is a 2-year grant, and our job over the remaining 8 months or so is to ensure that at the end of the day, at the end of the project, everyone at Horn Point Lab and beyond can access the data to conduct their own research with it. Finally, I'd just like to I just want to encourage anyone interested in this program to consider applying. I say that because the NSF program manager is on record stating that in the last year and a half, the number of submitted grants to this program has fallen precipitously, such that Meredith's grants that satisfy these programmatic criteria really have a high chance of getting funded. So in an attempt to capture the cultural zeitgeist of the moment, I have indeed wordled the outline of this talk. So the first order of business is to show you where PhytoChop is located. Next, I'm going to go into quite a bit of detail to describe the tools, both physical and cyber, that constitute the Coastal Observatory, and discuss some of the decisions that went into its design. A large part of the securing this grant was in our ability to justify its existence. And as I said earlier, its ultimate success will be in its ability to foster all new kinds of science. Therefore, in the sort of second half of my talk, I'm going to put forth a few ideas of how we think PhytoChop can help address both fundamental and applied science as it pertains to phytoplankton ecology and water quality. I will wrap up this talk talking about the remaining steps that we need to accomplish. And I've taken a little bit of liberty in this talk, made it a little slightly shorter than a normal seminar. So I'll be sure not to cut into anyone's lunch. So yeah, that was a bit of a forced wordle, but there you have it. So the first thing is where? The Coastal Observatory is located on the Horn Point campus at the end of our research pier. I've really come to appreciate this locale. It's one of the most beautiful places on campus, in part due to its panoramic views of the Choptank River. Currently, you go out there now, there's abundance of waterfowl in the area. So as shown on the picture on the right, at least from the outside, the observatory doesn't look like much. So what you're looking at is essentially a corrosion-resistant telecommunications enclosure. And for scale, it's about 6.5 feet tall, 5 feet wide, and 3 feet deep. So I can sort of just fit in there and fiddle around with equipment. One of the key things about it is that the unit has a programmable heating and air conditioning unit. So just by going out basically once a month, we are able to program this unit so we can keep everything near ambient water temperature and within the operating range of the various instrumentation throughout the year. I want to give a quick couple shout outs about this. First to Ralph Kynes, who's a member of our maintenance staff here. A few days after this unit was delivered and before any weight was added to the inside, the unit itself is just fairly thin aluminum. We had a strong nor'easter. So without any prompting, Ralph was kind enough and smart enough to secure the unit with some taut guy wires that you can sort of barely see running from the top of the enclosure. I'd also like to thank Jack Seabreeze, another member of our maintenance staff, who manufactured a mount that allowed us to move an unused davit from the boat dock over to the research pier, shown here to the back and left of the enclosure. And finally, to thank Steph Alexander and the Oyster Recovery Partnership for giving us some real estate at the end of this pier, as the enclosure is slightly in their way. So once you swing open the doors, you get a better sense of everything inside and how it's all connected. So if you're like me and are one of these type A peoples who get anxious at the sight of messy cables behind your desktop computer, I invite you to come down to PhytoChop and behold the ultimate test in cable management. Fortunately, as a telecommunications enclosure, it came with server rack rails, which we use to mount equipment, fixed shelving, as well as a lot of marine plastic. And what you don't see is there's a little divider down the middle of the enclosure that sort of helps us segregate electrical components on the left-hand side of the enclosure from sort of the more flow-through plumbing that's pretty much contained to the right-hand side of the enclosure. So I don't have an individual slide on electrical components. So just to briefly say that we have AC power running into the unit, but most of the equipment actually runs off either 12 or 24 volt marine batteries that we keep charged with an intelligent smart charger. The purpose of this is in the event of a power outage, the Ethernet, there's Ethernet down there as well, and critical equipment can remain powered and operational during any power outages. I've also hooked up an extra power surge converter that Kurt Flores donated to a laptop computer so that in the event of a power failure, I get an email. We've figured out pretty soon that this was necessary, as power often seems to get tripped down there just momentarily, presumably due to the cycling of a generator down at the pier. So in the following slides, I'll delve into the main pieces of equipment, but I just want to point out that though it may not look like there's still room for growth, we purposely bought a large enclosure that we needed to accommodate for the purpose to accommodate more science. In fact, what you mostly see on the right-hand side of PhytoChop pertains to Laura Wiltsey's MSc experiment, where she's installed a small enclosed flow-through chambers that house either oysters or just water, which serves as a control. Each of these chambers is equipped with a fluorometer and oxygen sensor to quantify oyster metabolism across all the environmental covariates provided by PhytoChop. And just circling back to cable management for a second, Matt and I made poor Laura go through several iterations of her setup before I was satisfied. Next slide. So everything but a wet lab's water quality monitor is ex situ, meaning outside of the water, and contained within the enclosure. Therefore, to draw water into the enclosure, as a group, we had some pretty vigorous discussions on how best to sample a tidal system. In the end, what we opted for is shown in a schematic on the left-hand side with a photo on the right. Obviously, the water is fairly turbid, so you can how do you sort of use a schematic for you to understand this. So for a few reasons, we ultimately reached a consensus to sample from 1 meter below the water surface, regardless of tidal cycle, and to have the inlets away from the pier as much as possible. Thanks to Bear, another member of our maintenance staff, we were able to achieve this by running tubing through rigid PVC that first runs down to the river bottom before running away from the pier. So there's rigid PVC with tubing going through it. Then affixing the end of our tubing with a small weight to 1 meter length of rope that's then tethered to a surface float. Part of the logic here is that during the summer, the oyster recovery program sort of circulates Chop Tank River through those large oyster tanks that you see out on the pier. So in this design is sort of our best attempt to stay as far away as possible from any sort of potential artifacts arising from that operation. We've tested a variety of pumps, and after going through a few impeller pumps that were probably a little too cheap as they quickly died, we're currently using a peristaltic pump to draw water into the enclosure, though we also have like a programmable diaphragm pump that could also serve that purpose. During the summer, we switch out the tubing about every few weeks just to prevent biofouling, and everything's sort of set up. So, kind of like a pit crew at a Formula One race with an extra set of hands, we can swap in and out the tubing within 20 minutes, so it doesn't really interfere with the operation of all the equipment inside. So once the water enters the enclosure, it first passes through a CTD, that's basically sampling every second, and then onto a small flow-through reservoir, which I'll show in a picture in a few upcoming slides. From this reservoir, we have plumbed in tubing that directly feeds the various instrumentation. And the first instrument I'm going to talk about is the most expensive and probably fanciest and interesting piece of equipment, and that is an imaging flow cytobot, or IFCB for short. Developed by Heidi Sosek and Robert Olson at Woods Hole, where our colleague Emily Brownlee did her PhD research, an IFCB is a rugged and waterproof flow cytometer, which additionally captures high-resolution images at a scale of about 3.4 pixels per micron. And that's done on the individual particles that pass through its fluidics. So on the figure on the left, much like a regular flow cytometer, this instrument works by drawing in a water sample through a programmable syringe pump and injecting the sample into a particle sheath fluid. These particles are then hydrodynamically focused to pass through a flow cell in single file where they're exposed to a laser light, 635 nanometers in our case. With regular flow cytometry, phytoplankton are detected and enumerated based on their autofluorescence after being exposed to this light, as well as how much this light sort of scatters off of a cell, which has been used as a proxy for cell size. So the IFCB does that, but additionally, what we can do with this instrument are trigger sort of thresholds. So for example, if a particle has X fluorescence and we think that's enough fluorescence that, that it represents a phytoplankton cell, we could tell the IFCB, okay, take an image of that. Nominally, most IFCBs are operated only to trigger the camera based on the presence of fluorescing particles, so phytoplankton. But we've set up ours a little bit different to not only trigger on fluorescence, so we're sure we're getting all the phytoplankton cells in the water, but we've also set up ours to trigger on particle size in order to potentially image other protists in the water, as well as things like detritus and what have you. The second photo, just to give you a sense of what the instrument looks like, is the IFCB in and out of its protective housing. This is a site, Emily, Laura and I know a little too well, as our IFCB, when it was delivered, was unfortunately delivered with some glitchy onboard electronics that had to be replaced, fortunately under warranty. Great news about this equipment. I've never used it before this project, but both Laura and Emily have a ton of experience operating IFCBs. And in addition to that, Emily Brownlee is also an excellent taxonomist. Before I get into some imagery, it seems like a good time to discuss why these measurements are valuable and sort of some of the advantages and drawbacks of identifying and enumerating phytoplankton from an IFCB as opposed to traditional light microscopy. So phytoplankton represent obviously a diverse set of photosynthetic protists and cyanobacteria that sustain the base of aquatic food webs, influence global biogeochemical cycles, and account for approximately half of global primary production, and are a large driver of water quality in environments such as Chesapeake Bay. Phytoplankton possess different, what we call ecophysiological traits. So this would include nutrient uptake kinetics, light harvesting efficiency, motility, and susceptibility to grazing. Ultimately, it's these traits and their interaction with the environment that really governs shifts in community composition through time and space. So observational methods often distill diversity down to phytoplankton functional types as delineated by morphology, pigment content, and/or taxonomic affiliation. So one potential drawback of the IFCB as an observational tool to sort of encompass this phytoplankton diversity is that it's limited to particles between 10 to 150 microns in size. Fortunately, as summarized in a recent Larry Harding paper shown here on the left, in Chesapeake Bay, the 25 most prominent taxa that were collected under the Chesapeake Bay Water Quality Monitoring Program have equivalent spherical diameters between 12 to 75 microns, so well within the operational range of our IFCB. The figure on the right is a time series of phytoplankton taxonomic data that was collected in the Choptank River, just a mile or so away from PhytoChop, as part of the Chesapeake Bay Water Quality Monitoring Program. For clarity and with the help of Sarah and Emily, we've coded individual species collected under this program into broader taxonomic groups. But really, the most important thing to note is that this time series stopped in 2011. Judy O'Neill, if she's on this call, can probably correct me if I'm wrong, but my understanding is that routine taxonomic monitoring in Chesapeake Bay is now much more sparse in space and time. So with that, there's obviously a need to collect this type of data. Also note that across this 20-year record, sampling was essentially confined to spring through the fall at either monthly or bi-monthly frequency, depending on the year. As I speak right now, the IFCB is running and it samples every 30 minutes. So obviously the instrument affords much higher frequency data than laborious light microscopy. And moreover, inside the IFCB, there's bags of bleach and biocide. So we can operate the equipment for approximately 6 months before having to worry about biofouling and then going out and just going out doing some quick maintenance and replacing the bleach and biocides. So it's sort of set it and forget it for a while. Okay, so on the left are some images from the IFCB taken out at the pier. Full disclosure, I'm not an algal taxonomist. I'm more of a phytoplankton physiologist, interested in phytoplankton ecology, but don't have the patience to take a water sample under a microscope, look at it and count it and learn how to do that. So the key to the IFCB data is really wholly in the data processing. And what I like to refer to as the digitization of taxonomic expertise, which seems to be a bit of a dying field of science. At this stage in the project, we're simply collecting data, but with Emily Brownlee, we're quickly working towards deploying a couple of key data pipelines. These pipelines already exist too, so we're not trying to reinvent the wheel. The first pipeline is an automated classification system that uses a supervised classification of images collected from an IFCB, such that you get a sample, you compare it to an image library where, you know, this is a Thalassiocyra or this is some type of dinoflagellate, and then the computer program basically identifies and enumerates phytoplankton taxa. Like I said, it employs a supervised machine learning algorithm, and that, you know, fortunately for us, these digital libraries already exist. And part of Emily's job in this project will be to further expand the training library with additional taxa as necessary. A lot of this work has been developed. A lot of the extant libraries have been developed in the Gulf of Mexico and as well as at Woods Hole. So there could be things in our water samples that are not well represented in these libraries. The other what I think is a really cool thing one can do with IFCB data and specifically the imagery is to extract geometric characteristics to estimate cell size and biovolume, as shown on the figure in the right-hand side of this slide. You know, going back to what I was saying earlier about distilling diversity down to phytoplankton functional types, one can imagine that combining image classification with the, you know, estimates of biovolume, you know, we can begin to group individual particles based on both their taxonomic affiliation as well as their size, so such as large diatoms, small diatoms, large dinoflagellates, etc. And by the way, that would be extremely hard to do with traditional light microscopy. So in order to sort of advance phytoplankton ecology and better understand things like seasonal succession and their drivers, you know, it's absolutely critical to have contextual environmental data. Beyond temperature and salinity measurements that are afforded by our CTD, we've also purchased an autonomous nutrient sampler from Green Eyes, which is a local company in Easton, run by a former HPL FRA, Vince Kelly. So as you can see in this photo, this unit consists of 3 measuring chambers, where each chamber, one measures ammonium, the other nitrate, nitrite, and the third soluble reactive phosphate. This instrument performs online colorimetric wet chemistry assays using standard methods. The round dials that you see in this picture that are just below the black flow-through cells are connected to an array of tubing that goes to a variety of bags, excuse me, that contain, you know, reagents, standards, waste, and of course the sample inlets. Unlike the IFCB, which is really designed for long-term unintended deployment, this instrument requires a bit more care insofar that, you know, reagents should be ideally made in the lab and replaced at what we think is approximately monthly intervals. Not shown in this picture is a small Raspberry Pi microcomputer that reads in the data and stores it in the cloud. Because the instrument is a bit more laborious, unlike the IFCB and the CTDs that are sampling at their maximum resolution, we've programmed these nutrient measurements to sample every 2 hours. The picture on the right, by the way, is taken from the back end of the observatory. At that bottom of the image, you can just see poking up is our peristaltic pump that's drawing in water through that whole sampling tubing I discussed earlier. And above it is this flow-through chamber I'd also mentioned earlier. I've included it here just to say that within this chamber are 2 small filters that exclude large particles before they enter either the IFCB or the new lab. The one in the new lab is shown with the red arrow. It's this little white block. And there's an additional filter for the IFCB that's just too tiny to see. So that's really just to not get anything large in there to disturb our measurements. Okay, the last major tool, and the one I'm most excited about personally, was ordered way back in September 2020, but due to a combination of COVID, supply chain delays, and actually Boris Johnson, it's not due to arrive until next month. This instrument is manufactured in the UK, and because of Brexit, the company has to now go and undergo some sort of rigorous compliance testing in order to sell to the EU market. And for whatever reason, I'm like, well, we're not EU, but they had to do all this stuff anyways, which really caused a lot of delays. So hence the Brexit Boris Johnson quip. Anyways, once it arrives, I've already got a home for it. So we should be able to quickly get it up and running quickly. So what is a BFRF and what does it do? So FRF is fast repetition rate fluorometry. It's a widely used variant of active fluorescence that essentially exploits the inverse relationship between photochemistry and fluorescence to measure something called the quantum efficiency of photosystem 2. This quantum efficiency of photosystem 2, you can think of it in a couple different ways. Probably the simplest way is sort of a metric of the photosynthetic capacity of a cell and how healthy it is, how quickly it's metabolizing. Over recent years, there's been really significant advances that have expanded the scientific utility of these types of measurements, including a lot of work I did as a PhD student in Canada and a postdoc in the Netherlands, working with people who manufacture these equipments, but also a lot of the experts in the field. So through a careful calibration procedure that I was involved with, these data can now yield instantaneous measurements of basically what's called the absorption coefficient of photosystem 2, light harvesting pigments, which is a mouthful, but it just essentially is a measurement of how much light phytoplankton are absorbing. When you couple that with how quickly, how efficiently they're able to take absorbed photons and process it through the photosynthetic light reactions, what that does is really enables accurate and autonomous estimates of photosynthetic electron transport rates, essentially a value which represents the energetic yield of photosynthesis, which ultimately fuels carbon fixation and growth in phytoplankton. This is a 4th generation instrument, and the great thing about these is consistently getting better and better. And with this specific one that we've purchased, it now possesses 7 excitation wavelengths, shown in the slide on the figure on the left, that sort of span the visible wavelengths. So by exposing water samples to these different wavelengths of light, what we'll be able to do is provide an independent assessment of both the physiology and biomass of different pigment groups. For example, phycocyanin-rich cyanobacteria that are, excited by yellow light versus, say, apridinin-rich dinoflagellates that are, more excited by blue light around 490 meters. I'm currently a member of a scientific committee of ocean research, or SCORE, working group that is really trying to standardize methods and data management. And as part of this group, I've had the opportunity to run this instrument in a lab setting pre-pandemic, of course, which is the photo I took on the right. Just to give you a sense of how it works, similar to the nutrient sensors, similar to the IFCB, we're going to be drawing water from that central cuvette. With all the instruments, I haven't said this, but obviously we're measuring precise volumes of how much the instrument is taking in, so that we can get really good volumetric rates of whether it be biomass or whatever we're measuring. So with the FERF, the water is drawn into a cuvette, which is then illuminated by a programmable set of LED lights. We're going to set up this instrument. There's tons of different ways you can set this up, but what we will do is program it to expose water samples to sort of incremental increases in irradiance in light, while simultaneously measuring and recording the sort of the photosynthetic quantum efficiency and the yield, the biomass yield of these different wavelengths. So these measurements are kind of analogous to traditional photosynthetic irradiance curves that people use to get at phytoplankton metabolism and primary production. But rather than gas exchange or carbon uptake, they're assessing electron transport rates, which is really the first and most critical step of photosynthesis. So alongside the IFCB, this gives us a good metric of what's in the water, insofar that it provides, sorry, I'll read that again. The instrument pairs nicely with the IFCB insofar that it provides an additional metric of diversity and biomass. And critically, unlike the IFCB, it gives us some good sense of phytoplankton metabolism. All right, I'm getting towards the end of the presentation insofar that when I was running out of time, so no more fancy pictures, fewer fancy pictures, a little more text heavy. But taking it all together, to the best of our knowledge, this will be the first observatory to sort of autonomously measure both phytoplankton community composition and physiology with contextual environmental parameters at high frequency. Taken all together, the resultant data stream will capture a quantitative description of phytoplankton community timescales at timescales that bridge processes from cellular division, so things that are super short, to seasonal succession, and with a bit of luck and some perseverance, longer timescales as well. I haven't really talked about the cyber infrastructure yet, in part because it's kind of underway, but data is currently being backed up daily to an offsite network storage device, especially for the IFCB. The IFCB has sort of an internal computer that's hooked up to the Ethernet that sort of sends out daily all the images and all the metadata associated with those measurements. And the plan is to periodically, approximately every 6 months, transfer this data to a long-term public repository, specifically NSF BCO-DEMO. The good thing about that is, really about all these methods, is in the last 3 or 4 years, and a lot of this has come out of a variety of different SCORE working groups, there's been really good standardization on how to best sort of archive and manage these data streams, with a lot of good manuals on sort of best practices. So it's still some work to do to finish off the grant. We've been fairly preoccupied with testing everything out, setting it up, making sure all the equipment's behaving as it should. We still need to do is sort of develop sort of the outward facing website, sort of to help with that. Maybe for those of you who are familiar with IFCBs, or probably have looked at a dashboard at some point in time. And what this dashboard is essentially like a web-enabled interface for you to see in real time the data that's being collected from the IFCB, and to go backwards in time as well to look at some of the imagery. And part of this is, developing, we're taking extant pipelines, but I'm sort of interested in also developing a few educational tools, which I think I'll talk about a little bit in the next slide. As part of the grant, we had to write a needs assessment. So basically things where we think the PhytoChop can sort of help advance science. So I'm just going to briefly go through three needs assessments. The first one is advances in ecophysiology through the lens of biogeochemical models. And I'm sure there's a lot of modelers on this call right now. So basically, accumulated over decades of research, we have sort of a broad but incomplete understanding of how different ecophysiological traits ultimately drive phytoplankton dynamics. This knowledge base is probably best exemplified by examining the structure of the multitude of aquatic ecosystem models that attempt to resolve phytoplankton community structure dynamics. For those of you familiar with these models, you'll know that often they distill phytoplankton diversity down to 3 or 4 functional groups, which I sort of talked about a little bit earlier, though more complex models exist. Each of these functional groups is defined by a set of traits that parameterize growth, loss rates within the model domain. And typically these traits include nutrient uptake kinetics, light-dependent photosynthetic rates, temperature-dependent metabolism, and susceptibility to grazing. So in the grant, we identified sort of 3 significant challenges that need to be overcome to better model phytoplankton community structure. And when I say we identified, we read a bunch of literature on people who do this for a living and basically took what they're saying in their publications. So these challenges are, first, there's a lack of data available to sort of calibrate and validate these sort of phytoplankton community structure models. The data is sparse. Second, the classification of diverse taxa into smaller sets of functional groups is often arbitrary. So you can have, different, within a diatom groups, you can have ones that do really good in the winter, ones that do really good in the summer, just as a simple example. And third, the parameterization of functional group traits is often based on really a limited set of monospecific culture experiments that are then broadly applied to diverse taxa. So someone will measure photosynthetic rates of a couple diatoms in a lab and then apply it to some of these models. So the PhytoChop Coastal Observatory has been explicitly designed to address these challenges. First, highly resolved taxonomic measurements will be of immediate value to, like, for example, the widely used Chesapeake Bay eutrophication model package. Like many models, the CBMP, this model classifies phytoplankton into one of 3 functional groups: spring bloom diatoms, cyanobacteria, and the third category being other diatoms, dinoflagellates. So the PhytoChop uniquely provides the breadth of data that allow researchers not only to better identify phytoplankton functional groups, but also through all these measurements that we're doing, sort of help sort of constrain and quantify some of these ecophysiological traits. With respect to sustained observations in estuarine environments, whereas we have a broad understanding of phytoplankton phenology in temperate marine ecosystems, our understanding and ability to predict community composition at sort of subseasonal to seasonal variability in coastal and estuarine environments is surprisingly still lacking. Obviously, these are highly dynamic environments where the rapid turnover of phytoplankton is a function of short division times, tightly coupled consumer-prey interactions, grazing essentially, and then, in addition to periodic seasonal and tidal forcings, the close connectivity to land means that phytoplankton dynamics are quick to respond to episodic events. So this figure on the right is taken from a Clorn and Jasby paper published in 2008. Sorry, I forgot to put the reference in there. What they did was basically compiled and analyzed long-term series of phytoplankton biomass, specifically chlorophyll A, from 114 different coastal sites, including what's labeled as number 16 is a site, the site that's just a few miles away from where we are now. Basically, what that is showing, if the x-axis is the degree of annual chlorophyll A variability, and the y is the seasonal chlorophyll A variability. And what you can see with the position of the Choptank River data on that graph is that, interannual variability can be as high as seasonal variability. So in that paper, the authors conclude their analysis with sort of two important questions that sort of to this day remain largely unanswered. First, how do estuarine and coastal communities adapt to irregular and unpredictable resources? And secondly, how can we extract, obviously climate change is a huge thing. Our Chesapeake Bay is warming up, things are changing. All of this is in the background of nutrient remediation plans. So how can we extract these sort of signals from phytoplankton observations in ecosystems where local scale processes potentially mask responses to these longer stressors, be it climate change or things like nutrient remediation. So we hope that the highly resolved measurements generated by PhytoChop will provide a detailed assessment of how coastal community consumers adapt to irregular and unpredictable resources. And, our hope is that, to have this thing operating for a very long time, and that sustained observations across years can help deconvolve signals of climate change from the relative influence of seasonal, tidal to episodic forcings. The last thing is education and training in autonomous sensor networks. Harnessing the data revolution is one of NSF's 10 big ideas. And across the spectrum of earth sciences, autonomous sensor networks are a key component of this revolution. And in addition, phytoplankton biomass and phytoplankton diversity are both essential climate and ocean variables. So going forward, PhytoChop will hopefully serve as a nexus for training and education for undergraduate students at St. Mary's University of College. Emily Brownlee's working on some teaching modules, as well as potentially graduate students here or really anywhere else. Because of the varied data that it'll collect, there's a wide array of educational opportunities that are available to look at either the physical or biological forcings on timescales that span sampling frequency hours, from hours to days to weeks to hopefully years. I'm just going to close off with something I heard at a conference probably about almost like a decade ago in Belgium. And it was a person saying the two rules of time series. The first rule is never start a time series. And the second rule is if you start a time series, never stop a time series. So that's our hope. And with that, I will thank you for your attention and take any questions.
Thank you, Greg. So for questions, people can put them in the chat or raise your hand. And I'm going to start with a question. So you have this 2-year grant, but if you want this time series to go on for the next 20 years, things are going to break and you're going to need more reagents for your nutrients and all this kind of stuff. So how does it transition from a 2-year grant to a 20-year project?
So that is an excellent question, in part because part of this grant was talking about that sustainability. And I was going to address it in this talk, but I decided not to. We're sort of still working on it. I think, at the scale of, and I've come around on this thinking only recently, in part hearing that this program is, this FSML program is not getting as many grants as you could, but on really the long-term timescales, I think, in like 10 years or so, some of this equipment might even be obsolete. So I think the long-term survivability is just to continue writing grants to replace equipment as needed. And for us to do that, the real order of business is to make the data widely available as possible, but also hope that people are using it and publishing it, sort of to demonstrate its utility. On the short term, we sort of have a plan that originally what we wrote into the grant was that with the IFCB specifically, and I think this is probably the instrument most people are interested in, we were going to allow for some offline analyses. So originally we were thinking of only sampling every 2 hours to be in concert with the frequency of the nutrient lab sensors. So within that interceding time, people can, for a fee, bring samples, do their measurements, and using that as a potential way to fund the thing. The operating costs, Syra will probably know a little bit better with respect to the reagents for the nutrient analysis, but yeah, this is obviously something that we have to keep in mind. We have some money left in the grant, so really the last thing we're going to do is to purchase a lot of stuff to make sure everything is fine. To make sure that we have everything we need for the next 4 or 5 years. So we've got like backup pumps, backup tubing. A lot of that stuff is taken care of.
Greg, hi, this is Al Place. The big problem is that 12 micron limit in the IFCB.
Yep.
You're missing Carlo, you're missing azidinium and so forth, which are the ones that are really impacting. I'm hoping that the resolution goes below that basically at some point.
Yeah, and I'm sure that I guess that goes back to Clara's question about sustainability. I mean, they're already making, from what I hear, the next generation IFCBs that might have slightly better resolution. So yeah, we're right now it's state of the art, so we're happy to have it.
Greg, I was thinking about that last slide that you showed about the interannual variability compared to seasonal variability.
Yep.
Now that one. And I'm wondering if that could be due to, or an easy hypothesis to test with your data would be whether or not that's related to short-term variations in river flow versus the normal dry versus wet year variability. And in a fixed location like that, you would see that as variations in salinity and nutrients compared to those interannual and seasonal variations in river flow. So there's a good record at the Greensboro gauge that you could look at for that to see if the reason that CHOP tank sits on that 1-to-1 line, as I believe it does, but I'm reading the graph right.
Pretty close, yeah.
Yeah, that would mean that the short-term variability in storm events and river flows are equally important as the interannual variability in dry versus wet years.
So I would counter that with an additional statement, Tom, insofar that the data that went into this was data collected by the Chesapeake Bay Water Quality Monitoring Program over the length of the record whenever this was published, I think in 2010, this paper, and it's based on monthly data. So that, that type of data may not capture, a lot of these small scale events that you're talking about.
Yeah.
So one of the things, with these sort of sustained observations is that we'll be able to sort of better understand, how sampling frequency potentially influences and informs these types of things.
Yeah. Really nice talk. I enjoyed it.
Thank you. Jacob, you have your hand up.
Yeah, very exciting system, Greg. I'm interested in sort of how one sort of your plan for analyzing all this data that's going to come out. I've got how you're going to start and share it, or maybe that's just beyond the scope of this. But also with the FRF and the IFCB, those data seem to be really complementary, but I'm not quite. I'm trying to figure out how exactly you're going to integrate them. Right? So the FRF gives you information sort of about physiology of some sizes of organisms, which are, a lot of them are cyanobacteria, which are smaller than what you're looking at, but maybe the data about the phytoplankton you can use to sort of normalize it. I'm just wondering how you get those to play together.
So let me first address the first question, because it's also pretty important as far as how we're going to sort of analyze the data. So our job is to curate the data, and it's everyone's job to analyze the data. I need to make that abundantly clear. As I said in the beginning, we're just caretakers. By the end of the project, we're a little behind on the outward facing part and making the data easily accessible. That's sort of the high priority going forward so that anyone could look at the data. With respect to marrying the IFCB and the FRF data, so this has never been done before. No one's ever really had these two instruments out working at the same time. This sort of gets back to Al's point that, we're missing a lot of, with the IFCB, really small particles. They'll show up in the IFCB as small fluorescent images. But I think with the fast repetition rate fluorometry, and specifically its ability to differentiate between different pigment groups, which is broadly correlated to taxonomic groups, I think it's sort of a second metric of what's in the water and the relative biomass. So that might help mitigate some of, if we have a bloom of small cyanobacteria that will show up only as small fluorescent cells in the IFCB, they'll really stand out in the fast repetition rate fluorometer as these, large biomass of fluorescing particles. But beyond that, I mean, there's just so many different ways to go with this data. But yeah, maybe it's another conversation at another time.
In the chat, there's a lot of talk about
Yeah.
tide gauges.
Yeah.
Look at the chat real fast.
Yeah, I didn't have a photo, but we had sort of this 1 in 50-year flood event. I think it was this past November that we went out to PhytoChop and just to make sure everything was okay. So we're sort of a little resilient for climate change with this current location. Question from Don. Hi, Don.
Hey, Greg, great talk. Thank you. You mentioned that the modeling component. So I guess I'm neither in neither area, but can you talk a little bit more about how you're going to incorporate the PhytoChop data in a Chesapeake Bay model?
So I'm not going to do it, but hopefully someone with a good statistical sense can sort of maybe potentially parse out the data insofar that, okay, at this time we've got, well, I guess two things. One, it could be used to help validate the data. So let's say these models are predicting, diatom blooms when we're seeing a lot of dinoflagellates, that sort of thing. But also I hope in the long term that it's possible to sort of, with those sort of comparative estimates, to potentially look at reparametrizing some of the physiology that's baked into those models, and as well as the different phytoplankton functional groups. So right now, I think the CBPM has 3 functional groups, but maybe with these sustained observations, we will see that 3 is maybe not wholly sufficient to capture the sort of underlying variability in the ecosystem. I don't want to say too much on this. I'm not a modeler, but I was hoping that someone like you and Raleigh or Ming, people with modeling or statistical skills, can eventually sort of take a look at this data and sort of tease apart some of these data streams in a way that's relevant to modeling.
Yeah, cool. Thank you.
Larry?
And thanks, Greg. So, hold on a second. So, this is really cool, but so when does filtration occur? Is there pre-filtration before it comes into the intake? Are you passing raw water through the YSI? What, you know?
So it's not a YSI, it's a, I didn't say it's a Seabird CTD.
Oh, it's a sequence. Okay.
Larry, if you have strong opinions on this, I'd love to know, because we've done a couple different things. One of the things I didn't mention, but there is also a SCORE working group on inline systems for like ferry boxes for ocean going marine systems that we're sort of following along. In those things that it's often common to see like these 100 micron sort of inline filters prior to filtration. We've done it a bunch of different ways in our testing. You know, originally I had a little mesh at the end of the tubing in the water, but that got biofouled pretty quickly. So right now, the only sort of filtering that's going on, nothing through before it hits the CDD, but before in that flow through reservoir, we have those filters that sort of remove large particles before they're shown to either the nutrient sensors or the IFCB, and eventually the BFRF.
How often do they have to be cleaned in your experience so far?
Like basically 2 weeks.
Oh, okay.
And it's not too bad. You get an accumulation of a bit of sediment at the bottom of it that I don't think sort of restricts the measurements too much. And because it's sort of a flow through system, the way it's plumbed, it's quite good at flushing itself. And I'll just repeat, I didn't mention it, but the FERF as well as the IFCB have really good, they're really set up for long-term deployments in areas like the North Sea that are sort of comparable.
What's the mesh size of those? I assume it's a screen.
Yeah, so the mesh size on the IFCB is, I think, 150 microns. I'm sorry, do you know the one for the new lab?
I do, and this is going to be a point of discussion, Tom. 20 microns, not 0.2. So the measurement for SRP is truly something between TDP and SRP. And our intention is to figure out that relationship. Yeah, that's sort of the state of the art with the in situ wet chemistry. Yeah.
I can see where that would be an issue for looking, well, not just for phosphorus, but also for ammonium, absorb the particles and so on.
Yeah, the biggest divergence seems to be for the phosphate, but yes. So the plan is to get a test. You know, because we're measuring it every 2 hours, we have many opportunities to build up relationships there.
Yeah.
Okay, well, we're at time.
Yeah, the end of the Wordle is lunch, so.
Yeah. Okay. Well, everybody, let's thank Greg again. It was really a great talk.
Yeah,
And,
come get your stickers if you want one. Thank you, Jamie.
Thank you, Greg.
Yeah, thanks, Mike. All right,
Okay,
bye, everybody.
See everybody next week.
Bye.